activated by SEC1. SEC1 is a gene that my mentor found that it affects linker cell DAT. It's a linker cell DAT promoter, right? Yeah, linker cell DAT promoter gene, yeah. And so we're looking for genes that are similar to SEC1 or that work with SEC1. There are two hypotheses. SEC1 either works alone to promote linker cell DAT, or it works in the MAP kinase pathway. And I'm going to show you MAP kinase pathway right now. This is the MAP kinase pathway, MAP3K, MAP2K, and MAPK. The kinases, they're the proteins that transfer phosphates to each other. Like MAP3 kinase transfers phosphate to MAP2 kinase, and it transfers phosphate to MAP kinase. And that's why they activate each other. It's like a chain reaction. So PMK1 is a special protein that signal many other proteins to be made, so animal can fight infections. So when there's an infection, there are many steps, and there's this group, NOSI1 and SEC1. These usually work together, and when these activate each other, and SEC1 activates PMK1, and that triggers immune response by signaling many other proteins to be made. And the same group is used in two different cells with two different functions. It can be used to trigger immune response, or it can be used in the nerve cell to establish the fate of the cell. Establishing the fate of the cell means deciding which receptors are expressed in the cell. So these two groups, these are the same groups. They work in two different cells, but they have wholly different functions. When calcium is signaled, it activates some proteins here, and they activate NOSI1, SEC1. Then SEC1 activates some other protein, but we didn't identify that yet. So my mentor wanted to know if NOSI1 or PMK1 affected linker cell DAT. And these don't have an effect, so SEC1 either works alone or works with similar proteins. That's what we're testing right now. So also there are many MAP kinase pathways, so we're testing if they have an effect on linker cell DAT or not. Our methodology is, first we developed a cross strategy to test different genes, if they have an effect on linker cell DAT or not. What I did most of the time was crossing worms to get a new strain of worms, and then we're going to score new strains to see if there's a linker cell DAT defect. And we can actually visualize the linker cell by using GFP. GFP is green fluorescent protein. You can see that it glows in the dark in a compound microscope. So this is linker cell here. And it actually glows under the microscope, so we can trace it. And if it's dead, there's nothing glowing, but if it's alive, it's not dead, then we can see it glowing. So that's how we know that if it's dead or not. And scoring means that I look through the plates and count how many worms are with dead linker cells and how many are with alive linker cells. So one of the genes that we tested was NTK1. So we wanted to track it, and DUMP5 is a gene that we use to track that. They're both in chromosome 1, so it's easy to track NTK1 with DUMP5. And there's a phenotype difference. Dumpy worms look like this. They're short, they can't move very easily, they're fat. And the normal wild-type worms, they're like thinner and longer, and they move more gracefully. So this is wild-type, and this is dumpy. The cross strategy we use for NTK1, it's a little confusing, but yeah. This is a gene, QIS56 and DUMP5 is another gene. So with the worms, there is usually only 2% males but we want males to cross, so we use HEM5. HEM5 means high incidence of males. So when we use HEM5 in a worm, there is high incidence of males. That means there's about 30% of males as opposed to 2% of males. And QIS56 is actually the gene for GFP. This is a gene that makes it glow, and DAPI5 is the gene that makes it dumpy. So we crossed QIS56 HEM5 males with DAPI5 hermaphrodites. And then we picked 6 GFP plus non-DAPI males from this cross. This cross right here. Hermaphrodites from this plate. This is continuing. Then we score the plates for the absence of dumpies. And if there are dumpies, we're going to pick 32 non-dumpy GFP plus hermaphrodites to individual plates and let them swap. Then we're going to look at the plates, the 32 plates. And we're going to score them for 100% GFP plus and 100% non-dumpy. If the plate has 100% GFP plus and 100% non-dumpy, that means we homozygous to QIS56 and NTK1. The results we don't have any complete results, but we have some results since it's been only 3 weeks. We're at the last step of the cross strategy right now. The cross is successful so far with NTK1. We're going to make a new cross with PIG1. We already started that cross, but it's a whole different cross strategy. Once the cross is completed, we will score for liquor cell survival. Our future work is we're going to finish the cross with NTK1 and look at the results. If the linker cell dies or not, and finish the new cross with PIG1 and QIS56 and 5 to test PIG1 gene. Also, we're going to test many more genes of the kinase pathway, test more MAP kinases. Kinases, yeah. These are my references. These are my acknowledgements. I thank Elise Blum, my mentor, the Hafsa, Dr. Sad, Professor Ross, HCS staff, and you. Any questions? Excuse me? As I, in the earlier slides I explained it helps with, if we understand the non-apoptotic cell death in linker cell, since C. elegans is a model organism, we can understand in other organisms cell death. Like, if a person has cancer and the tumor is growing continually, we want the cells to die and be normal. But first we have to learn how cell death works, and C. elegans is a model organism in that sense. So we're looking how cells die in worms to figure out how they die in humans. Any other questions?